Welcome back, everybody. The Oscars are coming up, and we hope you're going to support the nominations. There were some great performances in that film. Well, we had a fantastic morning. I certainly learned a lot. Interesting presentations, great information. Collaboration seems to be a developing thing. And now we're moving on to our afternoon sessions of the second part of day 2 of Analysis for Authenticity. We got 3 more exciting speakers to hear from before the Q&A panel. We've got a great discussion. This is your opportunity to ask a group of experts to answer the questions that you've already had in the back of your mind, or maybe to confirm some of your thoughts. So please make sure those questions come in. I'm really looking forward to that panel. So under the collaborating theme, what we're going to do now is have a speaker who is going to tell us about the global pioneering food industry intelligence network. Ruth McDonald is a board member of FIN, and also she is the corporate service director of Morrison's. More importantly, Ruth is an old friend and colleague of mine, and it's great to see you. We both used to work for the same retailer, so lucky to have you here, Ruth, and great to see you. And you're going to talk about the remarkable thing, which when I do my global travels, Ruth, people don't understand Finn. They can't believe there's that level of collaboration. So it's a really a thing that globally people respect and still don't understand why people are collaborating. So maybe you could give us a clue about what Finn is and how it works. Ruth.
Absolutely, Sterling, and thank you so much for having me come along to talk about the great work that we're doing at Finn. And as Sterling says, I'm Corporate Services Director at Morrison's. I look after all things safe, legal, quality and sustainability in my day job, but I'm also on the board of Finn, and it's in that capacity that I've come to talk to you today. So the Food Intelligence Information Network, you've heard a lot today and yesterday indeed about the risks that we face as a food industry, and that's a collective risk that we face, and there's a huge amount we can do to collaborate and work together to mitigate that risk. And what I'm going to talk to you about is our FIN model, that model that we're using to actually drive action and take tangible actions in the face of those risks that are in front of us. And FIN's whole reason for being is really about how we work to enable data and intelligence insights, to share them, to target action, and that might be singular action at a business level, or it could be collective action, and I'll talk to you a little bit more about some of the collective things we've done there, to both identify risks through data analysis and sharing testing data, and to also take action and to mitigate those risks collaboratively. We're very much a UK-based framework, as Sterling touched on. We are getting some global admirers now, but this is very much a UK-based framework, albeit our members represent a huge global supply chain. There probably isn't really a corner of the globe that our members don't source from, and we source across our membership a huge and diverse range of products and ingredients, everything from teff and the latest health food grains to flour, fruit and vegetables, and the standard things you would expect our businesses to source. And Finn is really a fantastic example of industry coming together to share data. As I said, its reason for being is really about safeguarding supply chain integrity based on shared intelligence and insight moving to action. And we've seen recently, the European study by the JRC make a recommendation to the European food industry that really is important to motivate an incentivized food industry to share data and have an independent data trust that stewards and governs sharing of data amongst industry members as well as competent authorities. And that's what we've developed here in the UK. So let me tell you a little bit about Finn's journey. And the journey began back in 2015, post Chris Elliott's post Horsemeat review of integrity and assurance in the food supply chain, he made a number of recommendations about the importance of data sharing and action and collaboration. And so 21 businesses took that brave step and moved forward together. Morrison's was actually one of those founding businesses that came together to step forward and do something really brave and work together collectively as an industry to harness this and create this network and framework. And so established in 2015, really is a safe haven to be able to share information and intelligence and be able to have open conversations about what those risks might be. And as Finn grew, we established our technical steering group. That's a really broad group. I'll tell you a little bit more about that group and its membership in a second, but really that's got cross-category expertise, cross-industry expertise and cross-ingredient expertise so that we can monitor and drive insight into action, really building on that expertise. And then in 2019, we made a massive move forward in signing information sharing agreements with the main industry regulators here in our market. So the National Food Crime Unit, the Food Standards Agency, Food Standards Ireland and Food Standards Scotland, so that we could have a more formal agreement around data sharing jointly. And that was an enormous move forward for us. And then again, moving our journey forward and bringing it more up to date. In the last couple of years, we've spent a lot of time and investment in our data platform. I'm going to tell you a little bit more about our data platform in a minute, but that's really allowing our members to, at the touch of a button, gain insight and information about how their supply chain compares with that of others. So it's a hugely powerful tool and one we are continuing to develop over the next few years. So how does Fin work? Well, essentially, we're bringing together information from multiple sources. We've got authenticity data coming in from our members. We're gathering intelligence from regulators, from border, from other information sources, and we're bringing all of that together. And when you think our membership is comprised of retailers, manufacturers, food hospitality businesses, it's a very diverse data set. And as I said, it's a very global data set that covers an enormous reach across the world, so gives us great intelligence. That information is gathered quarterly and it's fed into Eversheds, who on our behalf anonymise that data. So it's completely anonymised, then it's aggregated and built up into reporting dashboards and insight. And then we'll go through that data, we'll add insight to it, and then we produce member reports. And we also offer dashboards for members to be able to search their own information against wider food industry. If I come on to Fin governance, clearly it's hugely important that we have the right governance in place. For the Fin network, excuse me 1 second. Apologies, the vestiges of an old cough. And that Fin board is made up of people from across retail, from manufacturing, and also our legal counsel from Eversheds. Additionally, we're delighted to have Saskia Van Ruth with us, and I believe Saskia spoke to you earlier today, and she advises us on direction, latest trends and intelligence, and really helps steer us as a board in the right direction. I also touched on our technical steering group, and this is a hugely important group because this is really our working and steering group, drives our direction, analyses our information, and guides our work. And so it's been really important that it's well represented in terms of the food industry. 17 of our member companies are represented there, and that comprises of 10 food manufacturers, 4 retailers, and 2 food service businesses, along with our independent expert, and it's chaired by the wonderful Debbie Barnes from Marks and Spencer's. And across those colleagues who sit in that group, we think very carefully about who represents those businesses in that group. So it's also based on the individual experience and knowledge of those colleagues, to make sure that we've got as broad a scope across ingredients, markets, and supply chains that we possibly can. And in addition, we've got Sophie Lawrence, who is our Fin Programme Manager. So if you want any more information about Fin post today, I'll be sharing Sophie's details at the end, and you can contact Sophie or Rachel Hackett, our Secretariat, or indeed myself. So moving on to talk about the model and how we share those insights. As I said, we've got a really broad membership, and you'll see from the group of companies represented there, the whole power, the breadth of our membership group. And that's hugely important because, again, as I said, we want to make sure we've got that real global reach. You'll see on the right-hand side there, some of the, and it's dummy data, but it shows the breadth of sort of reach across the globe of our members. So hugely, hugely important. And the whole purpose of Fin is based on this power of sharing, this ability to arm businesses with the ability to connect the dots between information and take that intelligence and turn it into action. Our members actually represent £180 billion worth of the UK food industry. And between them, they conduct 50,000 tests or analytical surveillance tests per year. That is a huge amount of testing. And one of the key things we found when we established Fin was that actually there was loads of testing going on, but no insight, no benefit of that collective testing. Lots of individual businesses off doing individual testing, many covering the same ground, areas of industry not under appropriate surveillance. And so that's really been the power of bringing that together. So that power of harnessing over 50,000 tests a year is huge. And if I give you a bit of an insight into the types of things that people are testing. So in Q3 this year, our members conducted over 7,000 tests on fresh meat, 2,600 on fish and shellfish, 291 on herbs and spices, and 275 on fruit and vegetables, which is a really interesting split because we can absolutely still see that hangover from the horse meat scandal many years ago, still in over-index really in meat testing. Seafood, obviously an area where we do have, as an industry, a number of concerns, but meat, we're still over-indexing in meat versus things like herbs and spices testing and fruit and vegetable testing. And again, looking at that data at a macro level allows us to go back to our members and try to direct and suggest where testing would be better focused based on intelligence. It also allows us to really think about and focus on the type of testing that people are conducting. I know Mary touched on this earlier, but we're seeing 65% of testing being around speciation, which I guess is no surprise when you think about the numbers of meat tests that are underway. 22% around substitution and extension of ingredients, and then about 12% on compliance with law, and then an 8% around geography and where items come from. We also can cut that data by test type, so we can see the vast majority of testing out there PCR. Again, I guess not a surprise given the over-index on meat and speciation testing. So we're getting some really interesting data at a macro level as well as at an individual company level. And what that's been driving us to do is think about how we bring our members together to do some collective testing. And so back in 2023, for the first time, we did some collective testing together, and we focused that on edible oils and did some work around olive oil, rapeseed oil, sunflower oils, and got a group of our members together to conduct that testing together, anonymise the results, and then shared that back for action. A number of investigations were kicked off as a result of the outcome of that testing. And I'll touch again in a moment on what's next for that collective testing piece, but we certainly think as a board that is a good direction for our members moving forward. So what do you get as a Fin member? Well, you get lots of things. Primarily, and I think as Morrisons are also a member, as well as myself being a board member, the thing we absolutely use the most of are our data dashboards. So I'll show you some further examples in a moment, but that ability for us to be able to take an ingredient or take a supply geography and understand what others are seeing in that area when we're making purchasing decisions or when we're reviewing potential incidents or reacting to intel is unbelievably powerful. It's a fundamental part of how we assess risk within our own business. So that, I think, for me as a member has been hugely important. But alongside that, the member events that we run, so we have an annual conference where we share all of the latest insights and we bring people together to focus and talk about what they see as being the next challenges and opportunities at our annual conference in June. And we also run a number of training events, which again have been hugely well received by our members. We provide the food forensics newsletter to our members monthly, and that's a key part of providing that insight, intelligence alongside test results, which is hugely powerful and something our members feed back to us on as being key in operating their risk assessment programmes. And then, as I touched on, this important piece of work moving forward on coordinated testing programmes, working together to assess risk, take action. Our members tell us that helps them actually reduce their amount of spend on testing, but still get the benefit of understanding risk and targeting action towards it. So hugely positive and beneficial. And this is just a couple of examples of what our members can see when they go into their dashboards. So this is completely dummy data. Don't write any of these numbers down. But these are the types of things that businesses will see when they're looking at how they're performing, how many items they've tested, how many of those were anomalous, what their global reach looks like for the countries and their supply chain. So hugely helpful, and also what categories of products that they've tested. And they can drill down and drill down and drill down into this data. And they can compare themselves against the Fin membership. So you can see here across the top, that'll be an aggregated version of testing. So all of Fin's members are testing this number of products with this percentage of anomalous tests. And then a business can see how their business compares, and that potentially they might have higher or lower numbers of anomalous tests when compared with the Fin membership. So this is hugely powerful data for businesses, and I could spend hours talking you through all the different types of dashboards and product information that is available. So what have we achieved as Fin? Well, we've brought together that broad spectrum of food businesses working together to help protect consumer interests and strengthen UK defence against food fraud. I talked about the online dashboards. They're now able to provide access to over 300,000 test results and enable that more strategic and targeted approach. We've got great recognition for our leadership in tackling food fraud. We get lots of queries globally and inquiries from global businesses about how we could help them establish the same thing in their local geography. And our members get real-time notifications on key issues from Fin members and regulators, which is hugely powerful. It's given a voice to the UK industry, and we've really established this very mature and sharing agreement with the regulators, who we've enjoyed a fantastic relationship with. So to finish, really, what I wanted to share was just what we see those strategic priorities being moving forward. So we want to continue to enhance our data quality and depth and insights, and we encourage other businesses to come and join us to really grow that database and enhance the quality of action that we take. We're also extending our Fin support and resources to the small and medium enterprise community. We see this as an area of risk within the food chain, and we really want to do our bit in helping those businesses do the right thing. We really see joint testing programmes and collaboration around those as a huge opportunity. We've just completed a joint testing programme on basmati, and we're just analysing the results and deciding next steps. And we're already scoping our next set of collaborative testing around red spices. We want to expand the coverage of our food groups and really to continue to identify opportunities for collaboration with partner organisations in the food system. So that's been a real sprint through Fin and who we are and the benefit of working with us. If you'd like to get in touch, here are Sophie's details, or you can contact me. And of course, please ask any questions you would like in the Q&A at the end. So I'll now hand you back to Sterling.
Well, Ruth, that was fantastic. Thank you so much for joining us and sharing Finn. I must say, Ruth, when you mentioned the horse meat scandal, a kind of shiver goes down my back. But it did produce, it must have gone down yours.
Mine too.
Yeah, the bad old days. But it did produce an amazing report. I remember Professor Chris Elliott's report from Queen's University. And when you looked at the pillars, now I could understand the National Food Crime Unit. I thought, yeah, that would be a success. FAN was a recommendation. I could see that being a success. I must say it was the Finn element that I thought that's not going to work in a month of Sundays. People will not have the sharing of that type of information. And I think it's just an enormous success. And you say it was brave and courageous of the members to share that information. So I think it is, as I say, a global leader in that type of field. I don't know anyone else in the globe that's doing it. And I don't think most of the globe, as I said, really understand how it works. They can't believe that it works. So congratulations to you and your board and to your members for doing an amazing job.
Brilliant, thanks, Sterling. And I must call out Helen Sisson, Paul Wilgoss and Paul Dobson, who really were the three people who drove that through the industry. They've done through an amazing foresight and vision. Thanks.
Great. Let's celebrate their work. It was an amazing job. Now let's move on. Thank you, Ruth. To our next speaker. We have two more speakers to go before we try to answer some of your questions. And we've got a great speaker now. And we're going to immerse ourselves now into the effective screening point of contact approaches for food authenticity. And we've got Stephanie Heaney here. Stephanie, great to see you. Welcome. And Stephanie is a research scientist at Bioanalytical, based up in Queen's University, I believe, in Belfast, doing some amazing work up there. And she's going to tell us about cutting edge methods to detect adulterated food and how the screening methods can be used in an official control setting. So you're doing some incredible work up there. The research is amazing and the products you're developing are really, really interesting. So I'm looking forward to what you're going to say, Stephanie. Over to you.
Thank you very much. Thank you very much, Sterling, for the introduction. I'll just share my slides. Hi, I hope everyone can see them okay, and I hope you can all hear me. Firstly, just hi everyone, and thank you so much for joining today. It's a great pleasure to be presenting to you all, and I've really enjoyed all the amazing presentations so far today. So just a quick introduction about me. So I was diagnosed with a list of food allergies at a very young age. And like anyone with allergies, it makes you question everything you're about to eat. Is this food safe? What's in the ingredients list? Is there any chance of cross-contamination with an allergen? So this made me so interested in understanding foods, where they come from, how they're processed, and what testing is completed to keep us safe as consumers. So my passion encouraged me to study a BSc in Food Quality, Safety and Nutrition before going on to pursue my PhD. So my project focused on rapid analysis of the chemical composition of black tea leaves using vibrational spectroscopy and chemometric techniques. And both of these degrees were completed within the Institute for Global Food Security at Queen's University in Belfast. So now I'm working for Bioanalytical as a research scientist, where my main role here is the development and validation of food authenticity tests using spectroscopy and chemometric techniques. So for those of you who aren't familiar with Bioanalytical, we are a leading food authentication solution provider. We develop industry-leading, cutting-edge food adulteration testing methods that can help protect the entire food supply chain against food fraud. So we spun out from Queen's University in Belfast 5 years ago after building up our food integrity capabilities in the Institute for Global Food Security with Professor Chris Elliott. Although we are now a private commercial entity, we maintain residence in Queen's University and we have access to their world-class labs. So my presentation today focuses on the effectiveness of screening and point of contact approaches. So over the last decade, we've witnessed significant advances in analytical technology, application and scientific best measurement practices for food testing, inclusive of food authenticity, quality and safety analysis. This evolution has been mirrored in the capabilities of market availability of point-of-contact approaches. So these are recognised as portable analytical instrumentation, which can be deployed at point of sample testing throughout the food supply chain, often by using miniaturised equipment that offer potential to screen samples quickly and cost-effectively. So as these are rapid methods, they are valuable for speeding up quality control processes. They can be used at many stages throughout the food supply chain to identify food authenticity issues and ultimately protect purchasers and consumers against. So that leads us on to the question, what point of contact technologies are used for food analysis? So the main techniques that we see are vibrational spectroscopy platforms, including near-infrared, Fourier transform infrared, and Raman spectroscopy. We also see spectral imaging platforms, including multi and hyperspectral imaging, and as well as biological analyte-based platforms. So NIR is the most common form of point-of-contact testing in food analysis, as this method has so many benefits compared to other technologies. So firstly, it's non-destructive method, so samples can be tested normally without sample preparation. The technologies are easy to use, cost-effective, and can be miniaturized, just like this device in the photo. Method can be transferred easily, and results can be produced in real time. This is a non-targeted method, and samples can be scanned within seconds. Point-of-contact testing is applied in quality and safety analysis through a range of various food commodities. So the main uses of point-of-contact methods are testing for allergens, meat and fish speciation, as well as herb and spice adulteration, which I'm going to chat to you in more detail in the next slides. So I'm going to chat through a review of the methods for the analysis of culinary herbs and spices for authenticity. So this is an FSA report that was published in February 2024. LGC and Queen's University collaborated in this really interesting project. So what were the main aims and objectives of this project? So there were two separate phases. Phase 1 involved a review of the methods for analysis of herbs and spices by establishing needs via 5 stakeholder category-based focus groups, a literature review to identify current and emerging targeted and non-targeted analytical methods for the detection, identification, and quantification of herbs and spices and their potential adulterants. An e-survey through the Food Authenticity Network, which involved collecting broader stakeholder views on available, emerging, and currently being used methods for identification and quantification of herbs and spices. Assessing the practicability of testing in labs where method of analysis requires the use of a reference database, and how different targeted and non-targeted methods might be used as part of a screening process to verify evidence of adulteration. Establishing the availability of reference materials and proficiency testing. Phase 2 involved understanding the needs of public analysts to enable them to implement new technologies which would improve, support, or increase efficiency in herb and spices authenticity testing. So herbs and spices are a commodity group that, as Silverani mentioned earlier, consistently appear in the top 10 commodities most reported as being adulterated. Due to the large variety of products that fall within this category, the complex global supply chains, and the commercial production processes, methods for verifying their authenticity or detecting fraud are not straightforward. So the focus on this report was for the methods of analysis of herbs and spices in their dried form for authenticity, with a focus on detecting deliberate adulteration rather than contamination. So stakeholder engagement revealed that with respect to authenticity, herbs and spices of most concern are oregano, followed by black pepper, and then chili powder, saffron, paprika, and turmeric were next, with all of them rated equal concern by stakeholders. It was noted in the report that authenticity issues most encountered were substitution, adulteration, and concealment. Authenticity testing of herbs and spices is a complex matter, and the analytical approaches used for authenticity testing are very much dependent on stakeholder category and the purpose of testing. The priorities for authenticity testing vary between different stakeholders, as do the herbs and spices of primary concern, the sampling strategies adopted and the analytical methods employed. So the literature review identified a total of 132 analytical techniques for herb and spice authenticity between 2015 and 2022. So spectroscopy-based, mass spectrometry-based and DNA-based techniques feature most in the scientific literature. Data from stakeholder focus groups, responses to the Food Authenticity Network e-survey and the literature review were compared and assessed to identify the techniques most commonly used for the analysis of herbs and spices for authenticity. So results have been presented by a word cloud which shows that NIR and PCR were the most commonly used techniques, followed by FTIR, GCMS, LCMS, microscopy, NGS, ASH, acid ash, ICP-MS and HPLC, which were all equally used by stakeholders. So the availability of appropriate reference materials was flagged by stakeholders and public analysts as a significant obstacle to authenticity testing capability. So there are currently no certified reference materials available, and there's a limited amount of quality control and botanical reference materials available. The challenge of obtaining traceable, internationally accepted reference samples for comparison remains one of the biggest challenges for food authenticity testing methods and is a barrier to the universal standardization of these methods. Regarding proficiency testing, 4 were identified who currently offer 17 different proficiency rounds, which cover authenticity, quality, and safety. So there are currently no proficiency testing schemes available for saffron or black pepper. The availability of authenticity databases was explored, and the study found that there are several open access food authenticity databases. These are large publicly funded programs. Larger herb and spice producing companies have their own within company reference libraries, and public analysts said they were willing to work with other public analysts to reduce setup costs, and they would welcome access to third-party data. So what do public analysts currently use for authenticity testing? They're currently using optical microscopy method, and this normally takes about 2 hours per sample. Public analysts said they welcome methods or technology that could increase the robustness of testing or reduce resource requirements, provided it is economically viable. They said they would welcome further training in authenticity testing of herbs and spices. They've indicated that optimally they require a rapid screening method to serve as a triage approach for the initial analysis of herbs and spices. So FTIR and qPCR-based methods represent those best suited to being transferred to the public analyst labs. FTIR was expressed as a preferred method mainly as this can produce results more rapidly. So currently there is a lack of labs, including UK official labs, that can provide accredited authenticity tests for FTIR in herbs and spices. Via Analytical currently provide FTIR authenticity tests for 21 herbs and spices, 19 of which these have been fully ISO 17025 UCAS accredited. So BIA is the only UK lab with extensive UCAS accreditation, accredited FTIR-based methods for verifying authenticity of herbs and spices. Utilising this FTIR screening method comes with many benefits, and like all methods, there are some limitations. So advantages are the availability to test rapidly with a small amount of sample. Results can be produced within about 20 minutes. It's a non-targeted method that is cost effective and also can be ISO accredited. So the limitations of this method include the fact that this is a screening approach and does require confirmatory analysis. The limit of detection is greater than or equal to 5%, which is quite high compared to some other methods. However, the report identified an agreement among stakeholders that the threshold for economically motivated adulteration is typically around 5% or greater, so there is no requirement to detect very low concentrations of adulterants. Also, an authenticity study conducted by Queen's University in 2016 actually found that commercial oregano samples that were adulterated by more than 30%, So if this is a more real representation of the fraud in the industry, the limits of detection for this method are definitely acceptable. There are also opportunities to improve model robustness and help decrease the LOD for this method by exploring different machine learning algorithms. So results from the report have demonstrated how public analysts expressed interest for a two-tiered testing system. So this would involve a screening technique to obtain an initial indication of the authenticity of a sample, followed by, if necessary, a more confirmatory technique such as GCMS or LCMS, and this would help validate any suspicious or non-compliant findings. So the report has identified many recommendations for future work. The first recommendation was to ensure the availability of commercially representative reference samples for herbs and spices, identified as highest concern with respect to authenticity. So having these would allow greater comparability between methods, techniques, and labs, enabling greater confidence in the results. The report also noted the addition of proficiency testing is required, especially for those herbs and spices that have been highlighted of significant concern, and including saffron and black pepper. As I mentioned, there are currently no proficiency testing rounds for these commodities so far. So this would enable, so if possible, the FSA could consider funding closed proficiency testing rounds for public analyst labs to enable them to demonstrate ongoing competence, and this will also assist them in maintaining their UCAS accreditation in core areas supporting the resilience of the UK official food and feed control system. So the report findings also recommend the development of guidance or regulatory industry code of practice for authenticity testing of herbs and spices, as it would provide greater clarity on the required standards for industry, commercial and control laboratories, and local and port health authorities. Public analysts could also benefit from support in many areas, including training on food authenticity methods, consideration of how public analysts can access specialised sample preparation equipment required to test herbs and spices using the FTIR method, and an assessment of the costs of implementation of new methods so public analysts can be supported. Regarding future research, so given the trend towards methods that use chemometrics and AI methods for data analysis, it is proposed that data fusion methods are understood, as they may need to be employed to enable a judgment to be made on the authenticity of a particular herb or spice that has been analysed by a number of different methods. So further work is required to assess the suitability of using NGS for the purposes of species identification and verifying the authenticity of herbs and spices. And as all public analysts have capability to undertake PCR-based methods, further work should be undertaken to identify relevant PCR-based methods for determination of herbs and spices. And the final recommendation is a rollout of the FTIR method to public analyst labs to enable them in the future to use a screening or point of contact approach for triaging official control samples. So in the next part of my presentation today, I'm going to focus on how the technology transfer of the FTIR screening method can be completed. So there's currently a trial ongoing that is funded by the FSA and is in collaboration between Public Analyst Scientific Services and BEA Analytical. So this study investigates if the ISO-accredited FTIR screening method for organo authenticity can be successfully completed in a public analyst lab. So this trial started at the start of the year in January and will be running for the next number of months. So this is an overview of the experimental design of the study. So the work carried out by PASS is presented at the top half of the slide, while the work carried out with BEA Analytical is presented at the bottom half of the slide. So the first step of the trial is the preparation of the oregano samples, and some were spiked with 5, 10, 25 or 50% olive or myrtle leaves. So these samples will be analysed by public analysts using their microscopy method. A part of each sample will undergo a milling as per ISO method using a ball mill and subsequently be scanned on the FTIR instrument at pass. So these milled samples, along with the unmilled samples, were shipped to Beta Analytical's lab for analysis. So when the samples were received by Beta, 45 of the milled samples were scanned on the FTIR, while the unmilled samples were milled before scanning on the FTIR. So the next stage will then be analysing the spectral fingerprint of the samples by our cloud-based chemometric models, which have been developed by a fully created and validated organo database. So just a bit about the trial progress to date. So what we know from our side, so the samples have been prepared by PAST. So these are the spike samples and the milled samples that have been shipped to Beta Analytical. The leaf samples have been milled at Beta Analytical, and the samples are currently being scanned on the FTIR. So then the next steps will be PAS and Beta will upload their FTIR spectra to our cloud portal. So what this will do is average the 3 replicates for each sample, process the spectra, and predict them against our organo model. So an authenticity score and a classification will be produced for each sample, and results will be produced within seconds. The optical microscopy analysis will be completed by the public analyst labs, and then finally we will do a comparison on the results. So we will consider if the microscopy results are comparable to the results produced by the FTIR method, and we look to see if results from samples milled by the different ball mills are comparable, and if the samples that have been scanned by the different FTIR instruments are comparable. So there are a lot of future opportunities. We really hope to complete this methods transfer with other herbs and spice commodities. So ideally moving on to black pepper, chili powder, saffron, paprika or turmeric before commencing to the other herbs and spices. So we really hope to collaborate and participate in other trials with other public analyst labs. These future trials will be extremely valuable to help us verify the transferability of the FTIR method to other labs. So just to conclude, point of contact screening approaches can be extremely effective for protecting the food supply chain as they produce results rapidly. Suspicious samples can be detected early. These methods are generally cost effective, and a larger quantity of samples can be tested in a shorter period of time. And the future work, so we recommend support and guidance for public analysts and also industry, increasing the availability of reference materials and databases for method validations, and the rollout of the FTIR methods, which has already started. So that just brings me to the end of my presentation. I'd just like to take this opportunity to thank the UK Department for Environment, Food and Rural Affairs, the Food Standards Agency and the Food Standards Scotland for supporting this conference, and a massive thanks to the conference organisers for their hard work arranging this event. So unfortunately I won't be able to attend the Q&A session today, but if anyone has any questions, please don't hesitate to email them to my email there on the screen, stephanie.heaney@beta-analytical.com, and I'll get back to you as soon as possible. So thank you all for listening, and I hope you enjoy the rest of the conference.
Thank you so much, Stephanie. Yes, we've already got some questions coming in for you, so we'll forward them on and perhaps we can get back to the people who've asked the questions. That was fascinating, Stephanie.
Thank you.
I'm just starting to get my state pension now. I got it last month, and it's made me very reflective, and I've been thinking of 45 years in the food sector. I'm just absolutely gobsmacked the progress that's been made in analysis, as you've demonstrated. 45 years ago, it would have seemed like science fiction, some of the work that you're doing. So it's incredible that you and people like you and your company are really at the kind of bleeding edge of analytical science, and the work you do is really, really appreciated. You just showed how valuable a role screening and POC methods can play in food authenticity analysis. So I look forward to seeing further developments from your company. You seem to be at every conference I go to, you'll stand, so I always come and speak to you all. Thank you so much for doing such an informative and interesting talk, Stephanie. Thank you.
Thank you very much, Shirley.
the very best. Bye.
Thank you.
Well, that was really interesting. And we come to our last speaker of the day, and a further interest for me, certainly, in the type of subjects that we're covering. Our next speaker is Gavin Nixon. Hello, Gavin. Can see you. Can you say something so I can check I can hear you?
Hi, Sterling. I can hear you really well. Thank you.
Right. Good to see you, Gavin. More interesting stuff on analysis. Gavin is the science leader in molecular and cell biology team within the National Measurement Laboratory at LGC, where we are all today, specializing in food testing area. Gavin is going to talk today about authenticity testing using next generation sequencing. Talk about new type of science fiction testing. Gavin, the sequencing like this, I think, would have been unheard of 45 years ago. So it's come on leaps and bounds. And again, another organisation is at the cutting edge of this type of technique. So, Gavin, we're very interested to hear you say about your talk about sequencing.
Thank you very much for the introduction, Sterling. I appreciate it. And also a big thank you to the conference organisers and also the previous speakers as well. It's been a fascinating, very interesting conference, so thank you. I'll just share my slides, so hopefully everybody can see those. Okay, and it's in presentation mode. Can everybody see that okay? Brilliant.
Yeah, that's fine, Gavin. Very
fantastic. Thank you very much. I'll just quickly introduce myself. My name's Gavin Nixon. I'm part of the biological metrology team here at the NML, which is hosted at LGC. And today I'll be talking about food authenticity testing using next generation sequencing. So these are the new kind of next generation technologies that are moving away from standard Sanger sequencing into the kind of very high throughput, massively parallel world of next generation sequencing. So just a quick overview of my talk. So I'll just initially talk about food authenticity testing, won't dwell on that for too long. Talk about kind of research to support NGS testing that's been funded within the UK, specifically the DEFRA FA0194 project. A quick overview of NGS technologies. What are they? What are the kind of key technologies involved in this? A little bit of the kind of outputs from the current project, which is the kind of landscape review. Also talking about harmonization and standardization. That's kind of critical to the uptake of these technologies across the kind of broad food testing community. Talking about some of the laboratory evaluation work we're doing, and then just wrap that up at the end. So food authenticity testing obviously aims to help ensure that foodstuffs that we consume are accurately labeled, meet appropriate quality and safety standards as indicated by legislation. Next generation sequencing, that is part of the analytical toolkit. It's increasingly being used within the kind of broader testing community. It has a lot of kind of pluses associated with it, such as being it's kind of non-targeted, it's kind of looking at kind of low levels, it's sensitive as well, and it provides a kind of reliable tool for food authenticity testing. However, there are always kind of cons, well, not cons, but let's say challenges associated with kind of new and emerging technologies, such as how is it being applied to food testing applications. And this is kind of highlighted the requirement for standardization, harmonization, which is the kind of theme for today's conference. So it's kind of ensuring that NGS-based methodologies are fit for purpose. And what do I mean by that? Kind of appropriately validated, they exhibit robustness, results and interpretation are standardized. So that's some of the things that are kind of due to the kind of way it's developed are not always available within the kind of broader community. So this is where the kind of research comes in. So UK government's funding research in this area to help support NGS testing. So as I said, the kind of variability of NGS results highlighted the need for harmonization and standardization within the area. So DEFRA commissioned and funded a project which is currently ongoing with supplemental funding from the Food Standards Agency, and that is DEFRA FA0194, which has the catchy title Harmonisation and Standardisation in the Field of Next Generation Sequencing, abbreviated as NGS, for the analysis of analysis for food authenticity testing. The kind of overall project scope is looking to support the development of standards and harmonizations, which is crucial to this uptake. Establishing the requirements for minimum performance criteria. So these are essential for helping develop fit for purpose NGS methods and workflows. It's exploring the scope and limitations of NGS and how it can be applied and how it can be providing solutions to help drive the uptake of this within the UK and the kind of broader testing community. So the project itself commenced in June 2023, and it's comprising kind of two main parts: a landscape review, that's to help kind of understand the area better, and then a kind of practical laboratory phase. The project itself kind of incorporates kind of broad stakeholder participation from NGS technology developers, foodstuff suppliers and manufacturers, obviously governmental and accreditation bodies. So I'll give a very quick overview of NGS technologies for those who are kind of uncertain of what they are. Next generation sequencing, otherwise known as high throughput sequencing, HTS, is a kind of massively parallel approach to sequence to generate sequence data based on a particular sample. So it's gone from first generation sequencing, where you kind of typically apply a kind of Sanger-based sequencing technology, where you would sequence a single sequence at a time, to something that can parallelly, like in parallel sequence, thousands up to millions of different sequences all at the same time. So it generates a huge amount of data. It's a very powerful tool for non-targeted screening approaches, and it's the way that a lot of kind of clinical diagnostics, kind of food testing is going, due to the kind of the benefits associated with it. Over the last decade, it's been increasingly applied to the food industry, and it's been both used in the qualitative, which is the kind of main usage of it, and sometimes in a quantitative manner, whereas occasionally the kind of interpretation has caused some issues downstream. Leading technology developers, but I'm not limited to, of course, include Illumina, which is one of the very big players, Oxford Nanopore Technologies, smaller ones within the UK such as PacBio, and then also another kind of leading provider is Thermo Fisher Scientific as well. These all kind of use kind of slightly different technologies. They have different features and characteristics, but they all fit within that NGS testing space. The actual instrumentation itself can be divided into kind of short read NGS and long read NGS. The great thing about short read NGS is that it's well established. It provides very accurate sequencing within a short amount of time with lower cost than traditional methods. And examples of those would include the Illumina and Thermo Fisher Scientific instruments, which you can see on the right-hand side. You've got the Illumina instrument in the middle, and then you've got the Gene Studio located on the top. And then you've got long read sequencing. So these are great for if you need to sequence short, but also kind of long molecules, DNA molecules, RNA molecules, and they're good for kind of identifying structural variants, and also for whole genome sequencing and such. So examples of those would include kind of Oxford Nanopore Technologies and PacBio, which can in some ways be considered as next generation, simply because they do kind of real-time single molecule sequencing. And the MinION instrument, if you can have a look in the bottom right hand corner, essentially you've got the laptop, and then right next to that on the left hand side is the instrument. So you can see how small and compact it is. So that's a true kind of point of contact testing device as such. So just a quick comparison of the different features and characteristics of the kind of a few NGS technology platforms out there. You've got them, as I said, from Illumina to Oxford Nanopore. They use a variety of different detection technologies, ranging from fluorescent to ion to electrical conductivity. Key applications, there's a large overlap between them, obviously, because they have similar applications. Some are slightly better at certain applications, but overall they can do everything that I've highlighted as a key application. Read length, that's where they differ. You've got the short reads, which will go up to 600 base pairs in length. Then you've got the longer reads. These are the ones that you get very long sequence data for, so up to something like 4 mega base pairs. So you can see that you're up to kind of large, very long DNA and RNA molecules with these. The outputs vary, but they typically generate huge amounts of data, gigabases worth, which is an enormous kind of bioinformatics requirement to analyze the data. The runtime for these type of instrument systems kind of runs from hours to days. It depends on your application. The kind of core benefits associated with these different platforms: high sensitivity, speed, efficiency, long read length. So there's various different kind of feature sets associated with different platforms, and challenges. You do have challenges. You do have things that need to be incorporated into kind of downstream kind of data processing, just to make sure that you compensate for that. So when you're using NGS through authenticity testing, as I said, it's been increasingly used. It's a kind of emerging technology, but it's been applied in multiple different ways across the food testing sector. So there are a variety of different NGS approaches, but they kind of broadly split into either metabarcoding or whole genome sequencing. And what is whole genome sequencing? That typically involves sequencing the entire gene that is present within a specific test sample. So that's essentially looking at every kind of part of the test material and then bringing back the sequence data based on that. The alternative is metabarcoding, where you're looking at kind of amplification of informative molecular markers. So these are ones that are typically kind of seen as universal markers, such as cytochrome C oxidase, kind of MAC-K, ones that you can use that are kind of, they show sequence variability between species, but conserved in certain regions. So they're essentially kind of short, standardized sequences that are used for taxonomic discrimination. Metabarcoding has been found to be a kind of well suited to the analysis of food-based products, and it's frequently been used within the kind of NGS testing approaches. So when I'm talking about barcodes, what sort of barcodes am I talking about? There's a huge range of barcodes out there. They kind of range from animal specific, plant, fungal, microbial as well. I'll just give an overview of a few of these. We've got cytochrome C oxidase subunit 1. That's an animal species kind of species specific barcode. What's the benefits of that? It's universally accepted. You can find it across a large number of kind of databases such as BOLD, just NCBI. There's a lot of information out there. It shows good variability within animals. However, there is limited resolution in some species, and also it's all dependent on what databases are out there. For something that's not as, let's say, highly kind of sought after or kind of studied, then you will have underrepresentation of those species. So that's something to take into account. Then for the kind of plant-based materials that you're targeting, you've got things like MAC-K. So what we're looking at is kind of mitochondrial, chloroplast-based conserved gene regions that you can use for molecular identification. So as I said previously, the project itself was kind of divided into two main phases. The initial landscape and review phase, which is essentially to establish where we should be focusing our efforts on, kind of looking at best guidance, best practice, and seeing what's the actual kind of research that's already been undertaken, what we can apply going forward to the kind of practical phase of the project, and to provide guidance in general as well. So the landscape review was looking at the kind of current applications, and also importantly the harmonization and standardization framework. So this is the part that's going to help kind of underpin kind of NGS based measurements and improve confidence with them across the kind of broader testing sector. So the review highlighted NGS platforms in common use within the UK, such as Illumina MySeq, Oxford Nanopore Technologies MinION, PAT BioSequel, and also the Thermo Fisher Scientific Gene Studio S5. NGS-based approaches are typically led by metabarcoding, which makes sense. However, there's a lack of consensus regarding quantitative application of NGS. So that's something that's up for discussion in the broader testing community as to the actual correct application if you're trying to use NGS for quantitation. Potential harmonization challenges. So this is what's actually kind of preventing its uptake and bringing in kind of uncertainty into this analytical technology. So it's the complexity, the diversity of food matrices. You get different levels of processivity associated with them. The inherent variability of NGS platforms use different technologies, different workflows, the bioanalytical components. Then you've got different protocols, and then also the need to comply with different regulatory requirements. There are a variety of different tools required to support this harmonization standardization framework, and they include kind of the obvious ones such as reference materials, databases, inter-laboratory studies, kind of MPT schemes and such, and development of international NGS standards, which is kind of crucial to the kind of broader uptake across the kind of international community. So when I'm talking about harmonization, standardization, what am I trying to say? So harmonization is essentially looking at test results, making sure that they're more comparable, irrespective of the analytical procedure. And then the standardization is looking at kind of applying those test results and then ensuring that they're uniform across routine measurement and traceable to a recognized standard reference material stroke SI unit. So it's the kind of international or kind of local standardization. So the framework itself requires multiple attributes. As I said, international standardization, you've got reference materials, quality control again, and performance assessment. The reason for the kind of performance assessment is that it helps in the method validation, the method development, and is crucial for kind of benchmarking and aligning kind of core analytical performance, where you're looking at key kind of measurement criteria such as accuracy and precision. Also, there are existing kind of areas such as NGS quality metrics, and they're key to help support this kind of performance assessment process as well. So NGS performance criteria, so this is kind of taken from the kind of classic method validation criteria. There's been a lot of kind of work in the clinical diagnostic sector, and they typically look at similar criteria such as trueness, analytical sensitivity, selectivity, and precision. It will sometimes be phrased slightly differently, but often they are referring to the same thing. So just to give an example of trueness, so you're looking at how closely relating how the closest of agreement between the observed test result and the accepted reference value. So that could be a reference value that's been generated using a CRM or a kind of validated method as such. And then these kind of minimum performance criteria, as I said, are kind of crucial to helping to underpin the harmonization and standardization. So in addition to these criteria, you've also got kind of QC metrics. So these are the type of metrics that you can generate as part of a NGS workflow. And when I say NGS workflow, I'm talking about taking the sample from DNA extraction all the way to, this is the kind of taxonomic classification. This is the species identification that we found within our test material. So the nice thing about kind of modern kind of analytical methods within the lab these days, especially in the genomic sector, is that you've got access to kind of quality metrics derived from kind of sample purity. So you'd use something like a SPEC, a spectroscopy, or a fluorometric system. You can look at DNA integrity. That's crucial for kind of PCR-based methods. Where you are looking at how intact is my DNA sample. The less intact it is, the more difficult it is to use PCR to amplify and generate these barcodes. You've got the library controls, making sure that the actual processes within your NGS experiment to generate these NGS libraries is appropriate and you're not seeing any issues. And then you're talking about areas such as post-sequencing, where you're basically taking a sample, run it on the NGS sequencer, and then you're looking at things like yield. What's the total number of bases, reads per run? What's the kind of coverage? So you're talking about things like sequencing depth. How many reads am I getting that align to your targets? Is it thousands? Is it tens of thousands? What's your confidence in that? And then there's also things like the actual quality of the sequencing itself. So on a base by base basis, you can see the kind of something called the thread quality score, or also known as the Q score, which is essentially telling you what's the probability associated with that base that it was incorrectly assigned. So if you've got a Q-score above 30, that's generally considered to be really good for most sequencing reactions, but that's also dependent on the NGS technology employed as well. So I've quickly given an overview of NGS standardization, harmonization, and I'll quickly talk about the challenges now. So I did allude to them earlier on. So they are due primarily to the complexity and diversity of food matrices. We've got very complex food chains, food supply chains, And we bring in samples from all over the world, materials from all over the world, bring them together and generate a food product. They can be very diverse, and also the kind of multiple ingredients. So in the old days, if you were just looking at something that was just some meat, something that you by intelligence knew it was just one particular meat type, you could just do standard sequencing. When you've got highly complex sample stuffs, food materials, you need something that will look and kind of analyze those multiple species presence within it, just to make sure it is what it should be. You've got kind of reference databases and barcoding. There is a kind of lack of comprehensive curated reference databases for food species. Some are overrepresented, some are underrepresented. So it's trying to get that improvement in the kind of reference databases. Regulatory hurdles. Different countries have different regulatory requirements for NGS-based food analysis. And then you've got cost and accessibility. Typically, well, historically, NGS has been quite an expensive approach to apply for routine testing. It's becoming increasingly cheaper and more accessible, but cost is a factor definitely in kind of cost-sensitive situations. And then there's also the continual kind of technological development. It's not stopping. It's just continuously evolving. New systems, new platforms, new variants, new chemistries, all the sort of things, new methodologies, which also kind of bring uncertainty, bring kind of difficulties in trying to harmonize and standardization. But there are support mechanisms, obviously, as well. So improved training and kind of guidance and such, kind of across industry. Access to databases, that's kind of crucial too, and it helps underpin the whole of the NGS measurements. Reference materials as well, such as certified reference materials, and developing kind of fit for purpose minimum performance criteria, just to make sure that they are appropriate for NGS-based food authenticity testing. So, excuse me, I'll just have a quick drink. So the kind of second phase of the project I'm just quickly highlighting today was the kind of laboratory evaluation. So that's ongoing at the moment. So we used the method validation guidance developed in the landscape review and applied that to develop our own kind of fit for purpose evaluation study. So we decided to go down the metabarcoding route because that was the most commonly employed within the kind of food testing area. And then looking at kind of high priority adulteration targets such as meat. So we kind of picked kind of beef adulteration, and then herbs and spices, and we've gone for the classic oregano because that's one of the high priority ones as well. And then trying to develop a method that we could look at assessing performance characteristics and helping design develop the appropriate kind of performance criteria as well. So as you would with any sort of kind of development, kind of validation study, we've kind of taken appropriately sourced materials, gravimetrically prepared admixtures, extract those from those, and use those test materials to assess NGS performance. So across our selected platforms. So for the platforms we're actually evaluating, they kind of cover short read and long read sequencing technologies, so we can capture the kind of pros and cons associated with those, or the kind of benefits and challenges associated with those. And then the workflow itself, as I said, kind of goes from DNA extraction, the experimental phase, and the bioinformatics. So it's a very complex kind of workflow with lots of areas where you need to kind of get a good grip and a good understanding of what's going on. So the performance evaluation, so I said we were using that to kind of identify and develop priority criteria. There's the classical method validation performance parameters, but also bespoke NGS criteria that we're applying to the evaluation as well. And then this is going to help us kind of assess barriers to the implementation and uptake of validation and NGS approaches. So the workflow itself, when you're talking about NGS, what am I talking about? So as a part of our kind of development phase, we're kind of looking at minimum performance criteria, as I've said. We're focusing on plant and animal authenticity NGS workflows, and then we're using a kind of mixture of short read and long read sequencing technology. So for the long read, we're looking at the MinION, and for the shorter read sequencing technologies, we're looking at the GeneStudio S5. So if we look at the kind of the workflow towards the bottom of the slide, you've got the kind of 5 core segments. You've got DNA extraction, that involves kind of sample processing, quality control of the DNA. You've got barcode PCR library prep. Then you've got the NGS itself, as in that's the experimental work, where you can pull the kind of library analysis, look at the quality metrics generated, and then you've got the kind of bioinformatics. So this is where you take your kind of raw data and you start processing it, filtering it, mapping it so that you can assign it to different species, understand the number of reads, as in sequencing reads associated with different species, to get an idea of what is present within your sample. And then finally, you've got your data analysis and reporting as you get with any kind of analytical technique as well. So I'll just quickly go into the summary. So next generation sequencing have emerged as a valuable and reliable tool for food authenticity testing. However, there are challenges associated with this, which are primarily due to the complexity, the diversity of food matrices. There's also the variability of NGS platforms and protocols, and then the associated regulations across the kind of international landscape as well. Potential support mechanisms include kind of improved training, access to databases, reference materials, developing fit for purpose minimum performance criteria as well to help underpin measurements, to help provide a more kind of harmonized, standardized kind of methodologies and such. And this is all kind of aimed at, as well as supporting the kind of broader UK kind of food industry, but also UK government policies as well, such as from DEFRA, who one of their priorities is kind of to support farmers to strengthen Britain's food security. And then from the FSA, one of their missions is to ensure food you can trust, and a vision for a food system that is safe, food is what it says, and food is healthier and more sustainable. So these type of the project work we're doing at the moment is to help them kind of support those kind of UK government aims. And for acknowledgement, I'd just like to thank my colleagues at LGC. So that includes Miguel Pediguero, Malcolm Burns, and then the kind of the project team at DEFRA. So that's Sophie Rowlandson and also Ocean Ellis. And also I'd like to thank Rachel Pointer at the FSA as well for all their kind of valued commitment and support to the project. So thank you very much to everyone for your time. And if you've got any questions, I've been more than happy to answer them during the Q&A. So thank you very much.
Thank you, Gavin. That was really exciting. The developments in your sector as well, just outstanding. And it's going to play more and more, I think, a bigger part in the work we do in fighting for food authenticity. And Gavin, you're going to hang around because we're coming to our Q&A now.
Definitely.
So we're all looking forward to that. I'm going to invite the other speakers to join us as well. I might be turning up as Mary. I haven't gone on the change. It says that my signal's gone down, so I'm appearing as Mary today. So I might make more sense. Mary's far more intelligent than I am. Well, could I ask you to put yourselves on the screen so people can see you? And unfortunately, Robin, as you can imagine, has been very busy today, so he can't join us, but we have had a number of really fantastic questions for him. What we'll try to do is get in contact with Robin, put the questions that you've asked to him, and perhaps we can put them on the fan site, the answers as well. He did do a fantastic opening session for us, I think, this morning. So I can't see any of the speakers online. Can anybody else? Or I'll try my best to answer your questions.
We're all here, Sterling.
Oh, great. I can't see you.
We're here on the panel on the right. I can certainly see people on the right. So we're here. So fire away.
Okay, right. So I haven't got you on the screen, but let's start off then with Ruth. Obviously, your presentation made a huge impact, Ruth. A lot of people complimenting you on it. And I think because of the very nature of it, people are trying to understand how it works, because by its nature, it has to be secretive in one way to maintain its integrity. Great presentation, Ruth, and so pleased to hear that Fin are looking to support the SME, small, medium enterprises, and indeed micro businesses, as well as the small businesses. Can communications more. Is there anything more you can share about what that looks like? So how are you communicating with the SMEs? How are you drawing them in? A sector that doesn't perhaps do much testing. What's your thoughts, Ruth, for the future?
Yeah, so look, we're looking at a number of ways of how we can support them and having conversations with a range of businesses around how do we sort of deliver publicly available information for them through our website in a very sort of low cost or no cost way. So we're just working through that. There'll be some announcements in due course about how we plan to do that, but it's, as I said, one of our 6 priorities for this year. Board are actually together in a couple of weeks' time to just kind of sign off how we do that. So any you know, kind of thoughts from the wider group here, very welcome on you know, how we might support those groups. But as I say, we're talking to a group of them. We've got some ideas about how we might do it. We're just kind of dotting the I's and crossing the T's on that before we kind of go public.
Well, that sounds really interesting. It's always a challenge, isn't it? That it's quite easy to speak to large retailers and industry and even the big hospitality groups. But when you get into that SME level, it's not easy to make contact with them and to really understand what their needs are. But it's great that you're looking at that sector. I believe, Ruth, that the small medium business make up the majority of manufacturers, hospitality sectors. So it's important to get to. Would you reflect on that?
Absolutely, they make up the majority of the kind of the food industry in this country, and also I think we see quite a lot of issues originate in that group as well. So I think it's the combination of the two that is the reason why we think it's so important to support those businesses. I think it's just a question of how, you know, because what we don't want to do is also provide a load of data and no insight, because that's not helpful either. So yeah, couple of good ideas that we've got. We just want to kind of nut out before we go public with.
Okay, thank you, Ruth. Just to confirm, Malcolm, are you out there? We've been joined by Malcolm, but I can't see you, Malcolm.
No, I'm here indeed, Sterling. Yep, pleasure to be here.
Welcome to the panel. Could you introduce yourself, please, Malcolm?
Of course, yes, no problem at all. Good afternoon, everyone. Just again to thank the organisers, Fan, and the sponsors for a very informative and enlightening Analysis for Authenticity conference across the last 2 days. My name is Malcolm Burns. I'm the head of the GMO analytical unit, but also the senior scientific advisor on food analysis at the National Measurement Laboratory hosted at LGC. I've been involved in supporting Gavin on the NGS front and the project he's just presented, and also all DNA related aspects, and because of that I rather barged my way onto the Q&A panel this afternoon. Thank you.
You're most welcome. Thank you for standing in at short notice as well. It's much appreciated. A question for you, Silverani. Are there examples of good data sharing even when the collaborating laboratories are commercially competitors? How were potential blocks overcome? So I suppose that's the centre of expertise that we have. Silverani, how do they work together, sharing what is not competitive, but perhaps retaining some of their intellectual property right?
I think it's tricky, and we have to be honest about this. If a commercial organisation has invested hundreds of thousands of pounds and even millions in creating a robust reference data set, clearly they're not going to give it away for free, because that's their intellectual property. However, we've been doing some work through the government chemists, working with DEFRA and FSA and FSS, and it's currently coming to a conclusion, which we hope to publish in a few weeks or months, hopefully, is recognising the fact that the commercial databases do play an important role in ensuring the authenticity of foods in the global supply chain, but how we can get access to commercial databases in cases of dispute. So I think that would be a good place to start, because often there are disputes and these often hit the headlines, and from a food business operator's point of view, you just get a negative result, or your food is adulterated and then it's splashed across the headlines, which, you know, is not a good strategy for anyone, certainly not food business. So in that sort of situation, we're hoping to intervene, and there's been a working group that's been led by Michael Walker and David Hoyland, with a group of experts involving statisticians and forensic scientists who do use multiple sources of data in order to give evidence to courts in a forensic situation. So that got us thinking, well, if multiple sources of data can be used in a weight of evidence approach, and applied in court situations in forensics where sometimes it's life or death situation, why can't we then use a similar framework for food authenticity? Now, food authenticity, can be life or death, because we know that food authenticity is closely linked to food safety, and there's lots of lessons to be learned. So they've come up with a really, really good framework how in a dispute situation, commercial laboratories may open the data sets up for a certain group of people, maybe the government chemists, maybe regulators. And if they did, what sort of questions are we going to ask? How many samples should there be in a database of a certain type for it to be fit for purpose? So it is a complicated issue, but over the last year, the working group have been really working hard, and they've come up with a framework and a list of questions where we can work with commercial database owners. And certainly it's been well received so far. And we're hoping that once that's published, that sets a framework out there so that people can then start using that in discussions before results become public and we get all of the headlines and the associated impact that people have to deal with.
Could I just ask a follow-on question? We've been talking to Ruth Silverani about SMEs. So that must be a challenge for FAN as well. How do you get out there? Because as well as doing the analytical work, you do a lot of work about sharing what good practice looks like, what tools are available, etc. How do you see that challenge as Ruth does?
It is a challenge for a small business, especially a micro business. Complying with food safety regulations is a challenge, especially when they vary in the UK. Now they diverge from Europe, and obviously across the world, we know that regulations are different. So I think Finn are absolutely right in identifying that SMEs need extra support. I think there's a huge number of food manufacturers in the world. Over 95% are SMEs, food manufacturing businesses in the world, and they clearly need support and help. So from a FAN perspective, that's why we firmly believe that all the information on FAN should be open access, freely available to any stakeholder in the world. So we don't have any firewalls, people don't have to pay anything, so that everybody gets access to the same information on good practice, well, ideally best practice, but sometimes good practice is good enough, so that they can raise standards and say, well, this is what other people are doing, so maybe I can just mimic that good practice. I don't have to reinvent the wheel. So hopefully they don't waste a lot of time. And in our food fraud prevention pages, we list to the global tools available to people so they can pick the best tool that's available for their business.
Thank
and
you,
I also just wanted to add that the food fraud reports partner project that I showed, I mean, those three organizations in theory are commercial competitors, but yet they've come together for the greater good and have shared high level data and for the benefit of all stakeholders, because that's one of the things that have been challenging us. People often say that we're in a high risk environment, or food fraud must be going up, but there was no real evidence that we could pin that data on. So the fact that those three organisations have seen what we're trying to achieve and come together and given that data in kind, I think is a real big testament to them in terms of data sharing and what we're trying to do in FAN, sort of debunk myths and suppositions and try and take it all back to evidence base. So there are good examples of data sharing. Databases are challenging, and because it is protected by IP, but we're hoping there are mechanisms by which people may grant access in cases of dispute as a first step, and then we can go from there.
I think we should celebrate that worldwide. I think certainly the Elliott report gave everybody a big, huge step forward, and I think we're seeing some of the benefits of that. And a lot of people I know working very hard to maintain that. I read Lewis Carroll to one of my grandchildren the other day, Alice Through the Looking Glass. It makes a great read. And there's the Red Queen there who's running but not moving. And when asked why are you running, the Red Queen says, I have to run really fast to stay in the same spot. And I sometimes feel that's the work we do. We're all running very fast just to maintain where we are now, fighting the threats. Great work done by the food sector. Saskia, can I ask you a question? I found this idea of this community that you're creating, this ecosystem, and the way that you kind of put it into that kind of diagram. I wonder where you see yourself in maybe 5 years' time. I mean, you're focusing on Europe, but we all know it's a global food supply chain. Do you have aspirations to spread outside Europe or get stakeholders from outside Europe?
For asking, Sterling. Of course, we deal with global supply chains, And initially, because this is funded by the European Commission, we focus on, say, the reception end in Europe. But that does not mean that the community does not involve other producers outside Europe, of course. You know, nearly all our spices come from, from outside Europe, for instance, and also a lot of other products, of course. So we focus initially, that's why it's called the European Food Fraud Community of Practice, on the products that are consumed in Europe. But that does not mean that the community is even at this stage not larger than that. We also welcome any stakeholder, all the stakeholders from other countries. And there are also some ideas to set up something similar in Asia, for instance, and connect so that we would have more hubs also in other parts of the world. And
What a
you're
fantastic
absolutely
idea.
sure it's a global problem. Yeah.
What a fantastic idea to have hubs around the world. The challenge for FAN and Silverani is always trying to make sure that we're not UK centric or UK or Europe centric, but trying to reach out to other communities. So what do you think your aspirations are then, Saskia? In 5 years' time, where would you like to be? Where do you want that community to be?
I would hope that this community will become a community that evolves on its own, like the principles in social sciences for community of practices, that there will be people, the organizations taking over roles. We start the community now with the group of people that's involved and anyone who is going to join us. But at some point, let's say the beast will get its own legs and should move on. And hopefully it is of sufficient interest of all the parties. And that's also one of our tasks, of course, that it will be able to survive after those 3 years.
A very good point to make, Saskia. I think it's a marvellous initiative. We've got a question here about the weight of evidence approach, and Malcolm, you've joined us, and Gavin, you might want to try answering this as well. Someone's asked, we heard a lot about the scope and benefits of sorry, my glasses are not very good, pro-demonic approaches on day one of the conference. Great that people are still around asking those questions. What about the benefits of other techniques and other approaches? How much is complementary and how much is either for a choice or a method? So what would be your comment on that, Malcolm? I hope I was clear enough.
Yeah, sure. Yeah, I can kick off on that. Thanks, Sterling. A really good question as well. I think as we learned from Stephanie's presentation as well, there's lots of other approaches available, not just proteomic approaches. So we've got the rotational vibrational spectroscopy approaches. So she introduced the near infrared, Fourier transform infrared and Raman there as well. There's spectral imaging platforms such as multispectral imaging, hyperspectral imaging, mass spectrometry, NMR, of course, the protein-based approaches, but also DNA-based approaches as well. And Gavin highlighted lots of the sort of scope and attributes associated with DNA approaches in the last presentation. I really like to think of these approaches more as complementary as opposed to being mutually exclusive as well, particularly in relation to sort of today's sort of theme, which is collaboration for fitness of purpose, really there. So, for example, the DNA molecule, it's relatively resistant to degradation and can enable analysis in raw, cooked and processed samples. Examples being GMO analysis, which use short PCR amplicons, and also Gavin talked about barcoding regions as well. There's shorter barcoding regions for NGS, which allow it to be applied to cooked and processed materials. DNA as well, one of the aspects is it's ubiquitous, so it's present in the cells of in all the cells of an organism, whereas, for example, proteins and peptides could be tissue specific and expressed at different developmental stages of an organism's life. Now, that's not necessarily a disadvantage, it's just a different facet, really. And that's something we should be looking at sort of in terms of weight of evidence approaches and capitalizing upon to get an analytical result, I think. Again, DNA mainly it can be used for quantitative purposes as well. You've generally got a stable nuclear copy number associated with the genome as well, and that helps particularly for food analysis when we're looking at compliance against a legislative or labeling threshold, for example, GMOs, durum wheat, and potentially basmati rice as well. So I really see all these approaches as not necessarily alternative ones, but sort of confirmatory ones as well there. And I think Robin May earlier in the day at the sort of a kickoff presentation as well, the keynote presentation, talked about ensuring food authenticity requires multiple approaches. And Robin was talking mainly about sort of national and international collaboration, but I think this also extends to analytical approaches as well. So with some notable exceptions, I think food testing would benefit from augmentation across different analytical approaches, proteomic, genomic, etc. There's some high profile exceptions, GMO analysis being one of those. The DNA is referred to in the legislation, in the internationally validated protocols, and affords full quantitation. But I think the real question really is, how do we ensure fitness for purpose? And Lucy and Sophie referred to this on day one about the analytical toolbox. There's lots of sort of options available, but it's really important in choosing a fully validated method. So evaluating the performance characteristics associated with a method in order to generate objective evidence of its fitness for purpose, talking about accreditation, etc., but also potentially going further than that as well, looking at maintenance of quality and competency in the area as well. For example, generation of reference materials, and even thinking about provision of proficiency test schemes, examples of external quality assessment exercises, for example, by our good colleagues at Axio, proficiency testing, and other service providers. So really, I think a sort of multidisciplinary approach, looking at a weight of evidence approach, building up the bigger picture from all the different analytical approaches there, really to support the fitness for purpose, and really sort of tying in with today's theme about collaboration to for a fit for purpose future, really. So thank you.
Very well put, Malcolm. I think one of the challenges is, we have worldwide experts here on analysis, but the group, your customers, don't normally retain that type of information within organizations, and sometimes I found as a food manufacturer retailer is trying to know what questions to ask, the right questions, and you guys can help with that, and then how to interpret the information and what are the right tests. So I think it's very difficult for almost the lay scientist who's not deeply involved in the analysis to understand what tests are appropriate for what type of problems and how to interpret the information. Is something that like that resonate with you? Do you find that in your daily work?
No, absolutely. It's making an informed choice on what the appropriate sort of analytical approaches are. What are the options there, but also what sort of questions should be asked. So, for example, if it's a very different question from is this soya adulterated with GMO Roundup Ready soya, as opposed to is this soya adulterated, or is there something in there that shouldn't be in there? They're quite different sort of analytical questions there. And it's also being able to interpret the end result and make an informed decision on that for the end user and for the consumer as well.
Absolutely, Malcolm. That certainly resonates with me a lot. Gavin, a question to you about technological advances. Crikey, I had a half a dozen of my own, Gavin. I found the future really interesting. And people are saying, well, the future is almost with us today, but people are interested, what's it going to be like in 10 years' time? Do you think that there'll be a next generation of analysis that's going to come out of the ether almost, that we don't even know that will be there? I mean, who would have known all the work that's going on genomes, etc., would it exist? who would have known about the gut microbiome 10, 20 years ago? No one taught that at university. So do you think there's a step change next generation? Is it possible to predict what? And can I just add to that? What's AI got to do with all this? Will you be replaced, Gavin, by an AI system that will develop all this type of work, and you can go sit on the beach all day?
There are AI tools out there already, and they are being employed for NGS-based sequence analysis, and also to develop the kind of the bioinformatics tools as well, which can be very complex, evaluating, validating those. So AI is going to be increasingly important, kind of part of the kind of NGS ecosystem, definitely going forward. It's kind of probably a little bit limited at the moment, but it's got a lot of potential, definitely. But we need to understand the kind of the actual what the technologies can deliver, what are their kind of feature set, are they kind of appropriate for certain applications but not best suited to others. Trying to understand and trying to harmonize, standardize, that's kind of crucial to everything, I think. So you improve the uptake within the kind of broader testing community, people understand it and trust it as a kind of routine testing tool as such. With regards to the kind of next generation, so we've gone from Sanger sequencing, which was first generation, we've got next generation, which was typically things like Illumina based, then you've got next generation. So we're up to like the fourth generation with things like single molecule sequencing. You've got the kind of real-time sequencing that you get from nanopore technologies, and also from kind of Pacific Biosciences as well, where you're essentially sequencing a single molecule. But that's not going to be just kind of the exclusivity of DNA and RNA, nucleic acid, also going to proteins as well. It's going to go down the protein sequencing route, whereas it's quite complex and expensive at the moment, but you'll be able to do essentially what you can do with kind of next generation DNA, RNA sequencing with proteins as well. I anticipate in 10 years' time, there'll be some amazing technology that looks like science fiction, but we'll be able to apply ourselves within the laboratories, hopefully at a point of test as well, the point of need. So as you can see with the kind of MinION instrument from Oxford Nanopore Technologies, it's a very small compact device which you can use in the field. You can go to the Amazon and things like that. You can go around the world, and as long as you've got a laptop and very simple laboratory preparation equipment, you can do your NGS sequencing there and then. The bioinformatics component, that's a bit more processing intensive. You probably can't do that on your computer. You need to send it to the cloud. So it's kind of linking kind of what we do within the lab to kind of cloud-based services, cloud computing, the interpretation, and that's where AI could come into the part. And I can see things like kind of integrated devices where you've got onboard sampling and extraction, feeding straight into the NGS device, and then giving you a result within minutes, hours, and such. So hopefully that's the way it's going to go. Technology is always evolving, as you know, Sterling, so it's not standing still. There's always that kind of push to be the next best technology out there, and also to kind of grab market share as well and get people using it and thinking, this is really exciting, this is what I need to apply to all my food-based testing. And it's bringing price down as well. Price is going down, and then that's improving the kind of accessibility. So as far as I can see, prices should go down per test, and the capabilities should increase as well. So that's hopefully the future that we see over the next 10 years.
Sorry, I was just going to say, I failed in getting my 3 sons to enter the food industry. I've got 3 grandsons, and I'm hoping, Gavin, the excitement that's going on in analysis will encourage it as a career. Sorry, Malcolm, you're going to say?
No, not till I interrupted you. I apologise. I just wanted to pick up on a couple of really good points that Gavin made on that as well. Again, I think, you know, the term next generation sequencing, it's a slight misnomer. As Gavin has said, we're already onto the sort of 3rd or 4th generation of sequencing instrumentation. I think that's why it's often now referred to as sort of high throughput sequencing or massive parallel sequencing as well. And as advances in technology and science continue to improve, you know, it's in the interest of NGS manufacturers to also keep pace with this as well. As we learned from Stephanie's talk as well, there's a push for miniaturization, for portability as well. There's real-time aspects, different technologies, read lengths, etc. But as Gavin said, irrespective of the new technology and the advances being made, I think there's the requirement for the application of sound scientific and measurement science and the infrastructure to underpin the application of NGS as well. And so we talked a little bit about the importance of validating methods, the need for reference material, key guidance, databases and standards as well. And I think the need for harmonization in this sector was also highlighted by DEFRA's AMWG technical steering group, which published an article on the government chemist website talking about guidance notes on NGS applications for food authenticity testing. And just going slightly off piste here as well, and acting as my own moderator, there was a great question in the chat at the minute, I think from Silvio, regarding, there's a range of availability of NGS instruments. Some are more available than others. How do you validate these? And I think that's a really good question, that. And I think one of the unique facets described in Gavin's presentation was the ability to actually challenge 3 of the main commercially available NGS instruments with a common set of samples under one roof, so doing an intra-laboratory validation there. But additionally, what we didn't want to do was, we're aware that there's a number of providers of NGS instrumentation on the market. The project did not seek to be prescriptive in terms of saying, this is the instrument you should use, this is the one something is validated on. I think here the project is really looking to provide guidance on the minimum performance criteria which must be met associated with any NGS workflow to help ensure fitness for purpose, and that therefore leaves it open to the analyst and the testing laboratory to choose their own NGS instrument as long as it fulfills some of these minimum performance criteria. And one final aspect on the AI, yeah, I agree with Gavin. It's certainly being used at the minute as well. In the EU and for GMO analysis and looking at predicting the presence of organisms and products as a result of new genomic techniques. Things are being developed in terms of data spaces, so integrating separate or disparate data sources and showing the interrelation between them from different DNA databases, then using data mining, so interrogating those large data sets to try to enrich for information of particular interest, and then also using iterative machine learning approaches, so really sort of scanning the data and getting computers to sort of learn for themselves and perform specific tasks autonomously, and being able to pick up on DNA sequence information which is indicative of a GMO or a potential NGT being present as well. So yeah, the future is here and it's something we need to be well aware of. Thank you.
Absolutely. Thank you for that insight there, Malcolm. Look, I've got 2 questions for you, Ruth. As you can imagine, lots of questions on the work that you do at Finn. And I'm going to ask you the final big question, Saskia, before I move on to Silverani, who'll do the closing comments. So, Ruth, just 2 quick questions. Are there any moves towards an international version of Finn?
Yeah, good question. And I think as you know, you touched on earlier, Sterling, it is a very admired model. We get a lot of inquiries from overseas. We've presented at a couple of overseas conferences as well. At the moment, we're very focused on our UK operations, albeit obviously those businesses have, as I mentioned, a very global reach, and we have businesses in the membership who have overseas sites and sites in other markets. So we don't have an intention to expand overseas ourselves, but we're very happy to help educate others on the model and help educate others to go on that journey.
Yeah, it would stop the incredulous looks I get when I try to explain what it is. People just don't believe it could work. So it would be a fantastic model to spread. Yeah. And then the last question someone says of you is, as Finn expands, how do you prevent dishonest companies from joining the club? So do you have a way of filtering who can be a member and who can't?
Yeah. That's a really good question as well. So yes, we do. So when people apply to become members, we have quite a set process and governance around allowing businesses to join. And then there are requirements for businesses who are members of Finn. So for example, they have to report the data quarterly. If businesses don't do that, then there are implications. And unfortunately, sometimes we have to take the difficult decision to cease membership. Obviously, that's not something we take lightly, and there's very clear governance around it, but we do need to obviously protect.
Yeah, a great organisation. You can see the interest that it generates from those almost looking from the outside in, fascinated by how it works. Thank you, Ruth. So Saskia, you seem to be the kind of center of this big web. So it's great that you're kind of bringing all these people together. So you've got this, I think, perhaps more of an insight into who the community is and what we could be doing together than maybe anyone else. So if I gave you a magic wand, Saskia, and said you could change one thing in the food sector to protect consumers from food fraud and to make sure we have authentic products, a difficult question, what would be the one thing that you would wish for?
Yeah, that's a difficult one. Yeah, of course, we would like everyone to be honest and to produce genuine foods. Unfortunately, that's an illusion. And as we all know, we are all dishonest at some point in time. If we think about whether we have told perhaps a small white lie in the past 3 or 6 months, most of us have to raise their hand. So I think we cannot eradicate that. Fraud is everywhere, not only in the food sector, of course. Better understanding of why people cross those lines. I think there's still some developments possible there. I would love to, to interview a lot of these fraudsters to understand their drivers and their behaviors. I think we could learn a lot from that as well, apart from that we try to protect the supply chains. But I don't think, although it would be if I could use the magic, I would like the fraudsters to disappear, perhaps all the liars in the world, but I don't think that's going to happen.
Well, great insight. As I say, that's one of my passions, is seeing how behavioural science can be applied to food fraud and to understand what motivates people. I've seen your experiment with students, which tries to encourage them to lie and to cheat, and how easy it is to get people to cheat, good, honest people. So that's a very interesting insight that covers the past 2 days. Thank you for that, Saskia. So look, we're coming to the end of the Analysis for Authenticity seminar, the inaugural one that we've had. A big, huge thank you to all our speakers for yesterday and today. Well, a marvellous sharing of insights. I certainly learnt a lot from the conference and I've enjoyed it as well. Thank you also to the audience for your attending. Posting questions in the QA panel is really important for us. We need your thoughts. Please use the whiteboard that you find in the chat. We're actively listening to you, the audience. We'd be grateful, very, very grateful actually, if you complete the short survey that is in the chat. We want to make sure that we're meeting your needs, and we're looking at doing this again next year. So we want to know what you want more of, what you want less of. Please do that and influence our next conference. There are recordings of the conference and copies of the presentation, which you'll find on the fan site at a later date. If you've got any parting questions in your head, we'd be fascinated to know what they are. What do you think the priorities are for the food sector when it comes to authenticity, research and innovation? And what do you want to see in the next 5 years? What do you want to think what the next 5 years could be? So as participants, could you please jot that down as well? It would be appreciated. It's been great fun and a great privilege to join you together as the chair today. It was just really stimulating. But now I'd like to invite Selvarani. Selvarani, you there to say a few words and to close the 2-day inaugural conference. Over to you, Selvarani.
Thanks very much, Sterling. I won't keep everyone for long, but just a quick sum up of the last 2 days' activities. It's been our absolute pleasure for FAN to host this inaugural conference, Authenticity for Analysis. Thanks to the audience, as Sterling said, over the last 2 days for attending and posing some insightful questions for our panel and posting their thoughts on the interactive whiteboard. Our thanks to the 2 chief scientific advisors for their keynotes, who both emphasised the importance of science and evidence in underpinning government policy and decision-making, and its role in supporting development of new technology and innovation, allowing us to address future challenges in the food system. DEFRA's Chief Scientific Advisor, Professor Gideon Henderson, highlighted the many projects commissioned under DEFRA's Food Authenticity Research Programme over the decades and how authenticity research helps to support a more secure food system, from checking for undeclared ingredients in products on our shelves through to detecting grain stolen from the occupied areas of Ukraine, which was launched just last month. FSA CSA Professor Robin May spoke about the many actors involved in the UK food law enforcement system and how collaboration between all parties, including the food industry, is essential. Professor May also highlighted how sampling underpins all of FSA's R&D and surveillance projects and noted how recent FSA authenticity projects, such as the Herbs and Spices project, have advanced our understanding of the issues needed to tackle food fraud in such commodities. Our speakers from day one explained untargeted testing and machine learning in engaging and understandable terms, which we're all very grateful for. And it's surprising to me, certainly, that we've been using AI since the 1990s without even realizing it. We also learned just how much authenticity research DEFRA and FSA is doing to try and stay ahead of fraudsters and to protect legitimate businesses and consumers. Today's speakers highlighted the importance of authenticity reference databases and datasets and how their reliability and access to authenticity data is a barrier to uptake and implementation of methods. And we all need to be greater and have greater and continued cross-sector collaboration to be more effective in detecting and preventing food fraud in the global food supply chain. A huge thank you to our chairs. Day one chair was Lucy Foster from DEFRA, and day two chair was Sterling Crew, as you've just seen, for doing an excellent job of chairing and keeping us on track. A big thank you also to the teams at DEFRA, FSA and FSS for not only supporting the conference, but for all their hard work in helping us put the conference together and doing all of the pre-conference planning. My last thanks goes to the fantastic fan team. That's John Points, Gary Bird, Maya, and especially Mary for managing all the organisation of the conference to ensure that these 2 days have gone just as smoothly as they have. Please do complete the very short feedback survey so we can continue to improve future events. And if there's anyone out there who isn't yet a fan member, I hope you'll see that it's very easy to join and it's free to do so. So I hope you take up the offer with the QR code that will be in the chat. Thanks again for attending, and I wish you all a pleasant rest of your days. Thanks for joining. Bye-bye.